Synthetic biology is a scientific field that aims to design and construct new biological systems that do not exist in nature or modify existing ones, utilizing principles from engineering, chemistry, and informatics. In this video, we will explore how scientists are employing synthetic biology for various applications, ranging from biofuel production to medicine. We will now delve into some examples of projects and research that are significantly transforming the field of biotechnology, opening new perspectives for the future, and even influencing the possibility of colonization on Mars. Synthetic biology emerged in the early 21st century as an evolution of biotechnology, with the goal of gaining a better understanding of biological processes and applying them to solve various issues from energy production to healthcare. One of the most promising areas of synthetic biology is biofuel production, offering renewable and less polluting energy sources compared to fossil fuels. Biofuels are derived from organic matter, such as plants, algae, or agricultural residues, transformed into liquid or gaseous fuels through biological processes. Ethanol, produced by fermenting sugar from sugarcane or corn, serves as an example. However, the current process is inefficient and consumes significant amounts of water and land. Hence, scientists are exploring ways to enhance ethanol production using synthetic biology. Strategies include genetically modifying yeast, responsible for fermentation, to produce more ethanol from various sugars, such as cellulose found in plant cell walls. Another approach involves creating synthetic bacteria capable of producing ethanol from carbon dioxide and sunlight, mimicking photosynthesis. These bacteria could be cultivated in bioreactors, closed tanks where biological reactions occur, without competing with food production or occupying large land areas. While synthetic biology's biofuel production can benefit the environment by reducing greenhouse gas emissions and dependence on fossil fuels, it also poses risks, such as the potential escape or contamination of synthetic organisms, leading to harm to biodiversity or human health. Researchers at the J. Craig Venter Institute are exploring innovative solutions in this field. The J. Craig Venter Institute, a non-profit organization dedicated to genomics and synthetic biology, was founded by scientist Craig Venter, renowned for sequencing the human genome and creating the first synthetic cell. One of their significant projects involves creating synthetic microorganisms capable of producing biofuels from renewable sources like algae, plants, or organic waste. The goal is to develop more efficient, cleaner, and cheaper biofuels than fossil fuels, contributing to reducing greenhouse gas emissions and ensuring energy security. To achieve this, the Institute utilizes DNA synthesis, genetic engineering, and bioinformatics techniques to modify or create new genes and metabolic pathways in microorganisms, optimizing the production of ethanol, biodiesel, hydrogen, or other chemicals of interest. After numerous studies, in 2010, Craig Venter announced the first synthetic cell capable of reproducing and expressing characteristics defined by synthetic DNA. Craig Venter's synthetic cell is a bacterial cell whose natural DNA has been replaced by artificial DNA, created in the lab from the genetic sequence of another bacterium. The team's goal was to create new organisms with useful biological functions, such as biofuel, vaccine, or drug production. While considered a milestone in synthetic biology, Craig Venter's synthetic cell raises ethical, social, and environmental questions about the limits and risks of manipulating life. Craig Venter's synthetic cell has potential applications in various areas, including biofuel production, vaccines, drugs, bioplastics, biosensors, and bioremediation. These applications are based on the ability to design and modify the genome of the synthetic cell to produce useful substances or perform specific functions. For instance, the synthetic cell could be programmed to convert solar light and carbon dioxide into ethanol, a renewable and less polluting biofuel than gasoline. It could also be used to manufacture synthetic vaccines, made from DNA or RNA fragments encoding parts of the virus or bacteria causing the disease. Another possible application is creating biosensors, devices that use biological components to detect specific substances, such as drugs, toxins, or disease biomarkers. Biosensors can be used for quick, accurate, and portable diagnostics, applicable at home or in the field. An example is a device created by researchers at Stanford University, utilizing a strip of paper impregnated with synthetic bacteria that changes color in the presence of arsenic, a toxic metal that can contaminate water. Synthetic biology can bring benefits to medicine by improving the prevention, treatment, and monitoring of various diseases. However, there are also risks, such as the potential misuse or malicious use of synthetic organisms for bioterrorism or altering human characteristics. Applications of Craig Venter's synthetic cell are still in the research and development phase, facing various technical, ethical, and regulatory challenges. Some challenges include ensuring the safety and stability of the synthetic cell, preventing its escape or contamination of natural organisms, defining the limits and responsibilities of life manipulation, and establishing standards and laws for the use and distribution of the synthetic cell. The benefits of research in synthetic cells extend beyond biofuels and biosensors. Another field that can benefit from synthetic biology is medicine, aiming to develop new treatments and diagnostics for various diseases. An example of synthetic biology application in medicine is the creation of synthetic vaccines. Synthetic vaccines, as mentioned earlier, are produced from DNA or RNA fragments encoding parts of the virus or bacteria causing the disease. These fragments are introduced into the patient's cells, which start producing the pathogen's proteins, inducing an immune response. Synthetic vaccines have the advantage of being safer, cheaper, and quicker to produce than traditional vaccines using attenuated or inactivated viruses or bacteria. An example is the COVID-19 vaccine developed by Moderna, utilizing the messenger RNA of the coronavirus to stimulate antibody production. Another example of synthetic biology application in medicine is the creation of biosensors, devices using biological components like enzymes, antibodies, or cells to detect the presence of specific substances, such as drugs, toxins, or disease biomarkers. Now, how could synthetic biology become the key to Mars colonization? Colonizing Mars is an ambitious project involving various technical and ethical challenges. Starting with the travel time. A trip to Mars can take from 6 to 9 months, depending on the planet's positions and spacecraft type. During this time, astronauts are exposed to high levels of radiation, increasing the risk of cancer and other health issues. Additionally, the journey is long and monotonous, affecting mental health and well-being. Imagine the astronaut team being the best individuals who got along well, resisted monotony, and are now in Mars orbit, ready to land. Then they face a new challenge. Mars has a thin atmosphere, making spacecraft entry and landing difficult. The atmosphere is insufficient to slow down the spacecraft, but generates heat and friction. Thus, parachutes, rockets, and airbags are necessary to reduce speed and cushion the impact. Landing also depends on factors like weather, terrain, and communication with Earth. The distance between Earth and Mars ranges from 56 to 401 million kilometers, causing at least a 4-minute communication delay. This means humans cannot rely on immediate Earth support in emergencies or doubts. They also remain isolated from family and friends, leading to loneliness and longing. As if the chaotic planet entry, isolation, and loneliness weren't enough, the astronaut team must face the hostility of the red planet, scarce in resources. For instance, the average temperature is 63 degrees Celsius or 81.4 degrees Fahrenheit, ranging from 143 degrees Celsius to 35 degrees Celsius or 225.4 degrees Fahrenheit to 95 degrees Fahrenheit. The atmospheric pressure is only 0.6% of Earth's pressure, preventing liquid water on the surface. The gravity is 38% of Earth's, causing muscle and bone mass loss. Radiation is high due to the lack of a magnetic field protecting the planet. Additionally, there are sandstorms, craters, volcanoes, and canyons, making the terrain uneven and dangerous. In summary, our astronaut team would be in a challenging situation. However, synthetic biology can be a key to Mars colonization by offering solutions to living in a hostile and resource-scarce environment. For instance, synthetic biology can help create synthetic organisms that use Martian carbon dioxide to produce food, fuels, plastics, and other useful products for humans. Synthetic biology can also aid in developing synthetic vaccines, biosensors, and medicines to protect humans from diseases and contamination. Moreover, it can contribute to scientific research on the origin and evolution of life in the solar system. Nevertheless, numerous challenges persist for the effective colonization of the red planet. Crucial advances in areas like space propulsion, environmental sustainability, life support systems, and space medicine still require significant research and development efforts. As we strive for this interplanetary future, it is imperative to meticulously care for our current planet. Mars colonization remains a distant reality, and during this journey, preservation and responsibility toward Earth are fundamental. In summary, synthetic biology emerges as a promising key to overcoming the intrinsic challenges of Mars colonization. Confronting the adversities of the Martian environment, including a thin atmosphere, extreme temperatures, and resource scarcity, manipulating synthetic organisms can offer innovative solutions. From creating microorganisms capable of using Martian carbon dioxide for producing essential foods and resources to developing synthetic vaccines and biosensors to protect colonizers from potential health threats. Synthetic biology proves to be a crucial ally in the journey toward interplanetary colonization. I am Samuel Euphrasio, and it's a pleasure to have you on Every Life Chronicles. If you enjoyed the video content, kindly consider subscribing to the channel. Your subscription means a lot to me, and honestly, I wouldn't know how to express my gratitude. Additionally, don't forget to hit the like button, as it not only brings joy to me, but also represents recognition for the effort and dedication invested in creating this video, which, I can assure you, required considerable work to produce. Thank you for your support and for being part of our community here on YouTube.